everybody. Hope you all are fine and enjoying good state of health. Today's topic for discussion is spermatogenesis. This is the third discourse in the series of lectures on gametogenesis. The participant at the end of this session should be able to describe spermatogenesis, its prenatal phase and postnatal phase. During postnatal phase, spermatid formation and spermiogenesis. Describe the basic requirement for normal spermatogenesis. Give the hormonal regulation of spermatogenesis. Describe mature sperm and tabulate the timeline of spermatogenesis. Give the biological characteristics of sperms in semen. Explain some abnormal gamete formation with reference to infertility and clinical significance. And finally, tabulate the differences between oogenesis and spermatogenesis. Spermatogenesis. It is the entire sequence of events in which primordial male germ cells, that is spermatogonia, repeatedly divide and differentiate into mature spermatozoa, that is sperms. This process begins at the start of puberty and continues into old age. For practical purposes, it is divided into two phases: prenatal phase, which comprises origin and migration of primordial cells, and then production of spermatogonia. Number two, postnatal phase, before puberty and after puberty. In after puberty subphase, two events: spermatid formation and spermiogenesis. First, the prenatal phase. During this phase, primordial germ cells, which are committed sex cells like cardiogenic cells, are formed in the caudal part of epiblast during second week of development. They travel through caudal part of primitive streak during gastrulation and reach wall of the yolk sac close to allantois during third week. During week 4, they start migrating through dorsal mesentery to the proposed site of gonad formation, that is dorsal wall of embryo, and they reach there by the end of 5th week. They continue increasing in number by successive mitotic divisions during migration and also when they reach destination site, and then they are renamed as spermatogonia. Postnasal phase and its before puberty segment. At birth, primordial germ cells, which are large pale cells, are surrounded by supporting cell, the Sertoli cells, which are resting on a basement membrane. This whole arrangement is named as sex cord. Sex cords persist throughout childhood until puberty. Before puberty, under the influence of gonadotropins, follicular stimulating hormone and luteinizing hormone released by pituitary gland, each sex cord acquires a lumen and is renamed seminiferous tubule. At this time, its wall consists of Sertoli cells with spermatogonia resting on the basement membrane and various stages of developing spermatozoa in the deep recesses on their surfaces. Now, after puberty subphase of postnatal phase, which is divided into two parts: spermatid formation and their maturation. First, spermatid formation, which is also named as spermatocytogenesis. At puberty, there are roughly 1 billion spermatogonia in both the testes, which are present in the basal layer of germinal epithelium, and several types of these cells can be distinguished. Number 1. Type A spermatogonia. The first group comprises dog cells which repeatedly divide mitotically and reproduce themselves through homonymous division so that spermatogonia population is maintained. Some of the type A spermatogonia do not pass through homonymous division, rather they follow the so-called heteronymous division, in which a new type of daughter cells are produced. Type A spermatogonia second group, which comprises of pale cells, which are unable to revert back and produce primordial spermatogonia. During successive divisions, each new cell is more differentiated than the previous one. This process ultimately leads to production of a new generation of cells, the type B spermatogonia, each of which by mitosis give rise to two primary spermatocytes. Each primary spermatocyte, the largest cell in the seminiferous tubule, after a prolonged prophase of 22 days, completes meiosis I and produces two secondary spermatocytes, which are about half the size of the primary spermatocyte. Each secondary spermatocyte undergo second meiotic division and give rise to two spermatids, which are about half the size or less than half the size of the secondary spermatocyte. Throughout this series of events, cytokinesis is incomplete. Hence, the newly produced cells remain in contact with each other through cytoplasmic bridges. Hence, progeny of a single type A dark spermatogonium forms a cluster. Throughout their development, spermatogonia and spermatids remain embedded in the deep recesses of Sertoli cells. Sertoli cells provide protection and nutrition and help release mature sperms. Now, summary of the spermatid formation. Primordial germ cells are spermatogonia stem cells by repeated homonymous type of mitosis give rise to type A dark spermatogonia, which can maintain the progeny of primordial germ cells. Now, type A dark spermatogonia by repeated heteronymous type of mitosis give rise to type A pale spermatogonia. Type A pale spermatogonia through repeated heteronymous type of mitosis ultimately leads to formation of type B spermatogonia. Type B spermatogonia by the final mitosis give rise to primary spermatocyte, and Primary spermatocyte is the largest cell in the series. It by meiosis I gave rise to secondary spermatocyte which through meiotic II division gave rise to spermatids. In the lower diagram, we can see the cells type B spermatogonium by mitotic division giving rise to resting primary spermatocyte which through meiotic first division give rise to secondary spermatocyte, and this cell by second meiotic division ultimately give rise to spermatid. Now postnatal phase after puberty's second part, that is spermiogenesis, which is also named as spermiohistogenesis. It is a differentiation or maturation process through which spermatids are transformed as spermatozoa and includes number one, nucleus condenses and form the head. Golgi complex gives rise to acrosome, a head cap that covers anterior two-thirds of the head of the sperm and contains enzymes which help sperm during fertilization process. Number three, centriole form the tail, neck and tail. Number four, mitochondria form the mitochondrial sheath covering proximal part of the tail and provides energy for the movement of the sperm. Number five, most of the cytoplasm is shed off. In human, the entire process of formation of the sperm from spermatogonia, that is spermatogenesis, lasts about 64 days. Spermatogenesis is highly sensitive to fluctuations in environments, particularly hormones and temperature. For an optimal sperm production, a certain milieu is needed, that is number one, temperature. Seminiferous epithelium is sensitive to elevated temperature and adversely and it is adversely affected by temperature as high as normal body temperature. Testicular temperature is 2 to 3 degrees lower than the body temperature, and this is achieved by two mechanisms. Number one, thermal exchange between testicular or arterial and venous blood streams. Number two, testes being located outside the abdomen in the scrotum. Number two, hormonal control. Initiation of spermatogenesis occurs at puberty because of interaction of hypothalamus, pituitary gland, and Leydig cells. And mainly three hormones are involved. Number one, testosterone is the major factor that regulates spermatogenesis. It is produced by Leydig cell in response to stimulation by luteinizing hormone from the pituitary gland. Follicular stimulating hormone fuels spermatogenic epithelium and stimulates androgen binding globulin receptors on the Sertoli cell surfaces. These are essential for concentrating testosterone level high enough to initiate and maintain spermatogenesis. Notable point, intratesticular testosterone level is 20 to 100 times higher than concentration in the blood amongst healthy individuals. Number three, luteinizing hormone. It induces proliferation and maturation of interstitial Leydig cells and stimulates testosterone production by them. Number three, slightly elevated pressure is among the milieu, and this is provided by taut tunica albuginea. Hormonal regulation of spermatogenesis. Male gametogenesis, unlike females, begins at the onset of puberty. Onset of puberty, it heralds release of gonadotropin-releasing hormone from hypothalamus, which acts on the anterior pituitary gland and release two hormones. Follicular stimulating hormone acts on Sertoli cell and result in activation and mitotic proliferation of previously dormant spermatogonia, which are converted to primary spermatocytes, and they then enter the first meiotic division. Second is luteinizing hormone, which acts on the Leydig cells of the testes and lead to production of testosterone. Testosterone with follicular stimulating hormone acts on Sertoli cells and leads to number one, up regulation of testosterone binding globulin receptors, which allow further stimulation of Sertoli cells, hence activation of spermatogenesis. Sertoli cells also produce inhibin, which causes spermatogenesis to slow down. If we concentrate on the right side of the diagram, there is a negative feedback system in the shape of number one, raised level of testosterone act on hypothalamus and anterior pituitary gland and inhibit release of gonadotropin releasing hormone, follicular stimulating hormones, and luteinizing hormone, which cause spermatogenesis to slow down. When sperm count is too high, Sertoli cells produce more inhibin, which inhibits release of gonadotropin releasing hormones and FSH, which cause spermatogenesis to slow down. If sperm count falls below 20 million per ml of semen, Sertoli cells stop releasing inhibin, which causes sperm count to increase. Male gamete, which is also named as sperm or spermatozoon, is produced in the testes and is expelled along with other secretions of male genital tract during ejaculation. It is a 60 micrometer long, a freely swimming, actively motile cell having three main parts. Number one, head, which is 5 into 3 micrometer in size. It forms main bulk of the sperm and contains a haploid nucleus. Its anterior two-thirds is covered by acrosome, the head cap, which contains enzymes such as acrosine, which help sperm penetrate into oocyte. Neck is 1 micrometer long and is very narrow part. Third part is tail, which is divided into three parts. Middle piece 5 micrometer long is covered by mitochondrial sheath which provide energy for activity of sperm. Second part of the tail is principal piece, which is roughly 45 micrometer long and is covered by a fibrous sheath. Number three part of the tail is end piece, which is roughly 5 micrometer long exoneme surrounded by small amount of cytoplasm and plasma membrane. Fibrous sheath of the tail promotes lashing movements which enables sperm reach site of the fertilization. Spermatogenesis timeline. It is approximately 64-day cycle that can be divided into four phases. Phase 1 comprises mitosis of spermatogonia, lasts about 16 days and culminates by the production of primary spermatocytes. Phase 2 comprises meiotic first division, lasts about 22 to 24 days, and during this phase, primary spermatocytes are converted into secondary spermatocytes. Phase 3 meiotic second division lasts about a few hours, and during this phase, production of spermatids takes place. Phase 4 is spermiogenesis, lasts about 24 days, and during this phase, maturation of the sperms takes place. Total duration comes to be 64 days. Biological characteristics of sperms in semen. First, the numbers of these sperms. Normally, 20 to 200 million sperm per ml of semen is considered to be the normal. If it falls below 20 million sperms per ml or less than 40 million sperms in an ejaculate, it is considered to be low. On the average, 100 million sperms per ml of semen is considered to be the normal range. As regards the structure of the sperm, up to 10% may be grossly abnormal. If this ratio is above 20%, this abnormality may affect the fertility of the couple. As regards the motility at emission, more than 80% of the sperms are motile. If more than 20% are immotile, that may affect the fertility. As regards the rate of the movement in the female genital tract, sperms travel roughly at the rate of 5 millimeter per minute, and about the survival time in the female genital tract is considered to be 3 to 4 days. Abnormal sperms and infertility. Three types of abnormalities of the sperms are identified. Number one, morphological, which are more common than oocyte and may affect either the head or the tail or both. As regard the head and tail, they may be abnormal in the shape of two heads, two tails, or an acrosome may be absent. Sperm may be of too large a size, that is the giant, or too small a size, that is dwarf. Sperm may be gianted. Second type of abnormalities are related to motility. For potential fertility, 50% of the sperm should be motile after 2 hours of ejaculation, and some of them should be motile after 24 hours. Third cause of abnormality is genetics. In this type, sperms may have abnormal chromosomal contents, but such abnormalities are rare as compared to the oocytes. Clinical studies relate mainly to infertility in male, and infertility is defined as failure to achieve a pregnancy after 12 months or more of regular unprotected sexual relationship. And this could be due to number one, oligospermia, that is low sperm count. And it is considered that less than 20 million sperms per ml of- or less than 40 million sperms per ejaculate after 72 hours of abstinence from sex. Second is azoospermia. Most probably, this is because of a blockage in the duct network of the male. Number three is lack of sperm motility, which is usually part of immotile cilia syndrome. A very interesting thing is that Mark Zuckerberg, the Facebook founder, one of the most successful entrepreneur on earth, is suffering from infertility. Now, oogenesis versus spermatogenesis. Oogenesis starts during fifth gestational week, whereas spermatogenesis, the actual process starts at puberty. Number two, process and duration. Oogenesis is a cyclical process and almost abruptly ends at menopause, that is, at the age of 45 to 55 years. Spermatogenesis is a continuous process of production of sperms. It gradually declines with age. Number three, motility of progeny. Oocytes are immotile, whereas sperms are highly motile. Number four, primary oocyte and primary spermatocyte progeny. During oogenesis, one mature oocyte and three polar bodies are produced, whereas during spermatogenesis, all four mature sperms are produced. Number five, size of progeny. Each oocyte is a large cell with three very small polar bodies, whereas all sperms produced during spermatogenesis are of equal size. Number six, hormones involved. During oogenesis, gonadotropin releasing hormone, luteinizing hormone, follicular stimulating hormone, and estrogens are involved. On the other hand, during spermatogenesis, gonadotropin releasing hormone, luteinizing hormone, follicular stimulating hormone, and testosterone is involved. Number seven, genotype. Oocytes are only of one type, that is 23X, whereas sperms are of two types, that is 23X and 23Y. Comments and suggestions will be welcomed. Thank you for listening and watching.